Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa. In today's video, we are going to take a look at photosynthesis. Photosynthesis feeds the biosphere. It is the process that converts solar energy into chemical energy. Only about 1% of the sun's energy is actually captured and stored by producers. In photosynthesis, energy of the sunlight is used to oxidize water. Electrons and protons are used to reduce carbon dioxide to glucose and process oxygen as a byproduct. We all know that plants need sunlight, water, and air in order to survive. But what exactly are the plants doing with this? Let's take a closer look at how photosynthesis works. So this is just an overview of what's happening in this process. So plants are going to use the energy from the sun, that solar light, in the presence of both water and carbon dioxide, specifically that carbon dioxide. The plant is going to take those in, and through that, it's going to generate glucose, which is energy or food for the plant itself. As that plant is working to make its own food in this process, it is also going to produce some byproducts we see here, which is water and oxygen. It releases that oxygen into the air, and then other living organisms can use that oxygen for cellular respiration. And in cellular respiration, that is going to generate carbon dioxide that gets exhaled out by living organisms, and the plants are going to use that. So there is this relationship between the plants undergoing photosynthesis, producing oxygen for other living organisms taking that oxygen in, and allowing for cellular respiration to happen. Now, in photosynthesis, the plants are taking in that carbon dioxide, that water, in addition to the solar light, that energy from the sun, in order to produce its own food. And we are going to continue on talking more about the process of photosynthesis. We can group living organisms based on how they acquire their organic molecules. Autotrophs are self-feeders. They sustain themselves without eating anything derived from other organisms. They are the producers of the biosphere. They produce organic molecules from carbon dioxide and other inorganic molecules. Almost all plants are autotrophs. Specifically, they are referred to as photoautotrophs because they use light as a source of energy to synthesize their organic molecules. Heterotrophs are unable to make their own food. They need to obtain their organic material from other organisms by eating. Heterotrophs are the consumers of the biosphere. Almost all heterotrophs, including humans, depend on photoautotrophs for food and oxygen. Before we take a closer look at the process of photosynthesis itself, let's take a look at the structure of a plant and understand where photosynthesis is taking place. The leaves are going to be the major locations of photosynthesis. Specifically, if we look into the leaf, we will find this area of tissue in the inside of the leaf referred to as the mesophyll. Within the mesophyll, we find these membrane-bound organelles that are called chloroplasts. So all reactions for photosynthesis are going to occur in this specific organelle that we find within the tissue in the leaf called a chloroplast. The stomata, or the stoma, that we find in the underside of the leaf is going to be the area where gas exchange occurs. So this is going to be the area where the leaf can bring in carbon dioxide, but also release oxygen into the atmosphere through these microscopic pores. If we take a look at the chloroplast itself, we'll see that it's a membrane-bound organelle. It actually has two membranes, an outer membrane and an inner membrane. Within the chloroplast, we'll see this area in the chloroplast known as the stroma. The stroma is a semi-liquid substance that surrounds the thylakoid membrane system. It contains enzymes that are required to synthesize organic molecules from carbon dioxide using energy from ATP. These reactions are going to be coupled with the reduction of NADPH, where NADP is the electron acceptor. Thylakoids that are found within the double membrane structure of the chloroplast are surrounded by that fluid, which is the stroma inside of there. So we see thylakoids that are in this area. And these are bilayer membranes that are continuous membranes with that internal membrane of the chloroplast. They are stacked in columns that are called grana. Chlorophyll, the pigment which gives leaves their color, their green color, is going to reside within the thylakoid membranes. So as we continue to talk about photosynthesis, you're going to see how this area is all going to play a role in how photosynthesis happens. When we compare photosynthesis to cellular respiration, we see that photosynthesis actually reverses the direction of electron flow. Photosynthesis is a redox process in which water is oxidized and carbon dioxide is reduced. Photosynthesis is also an endergonic process. The energy boost is provided by light from the sun. We take a look at photosynthesis again. Remember, we're going to use energy in the presence of both carbon dioxide and water to produce glucose and oxygen. Now, in this process, which is a redox reaction, we are going to have carbon dioxide that becomes reduced to glucose and water that is going to become oxidized to oxygen. The equation for photosynthesis makes it seem like a pretty simple process, but it's actually quite complex and consists of two different stages. There are the light reactions, which is the photo part of photosynthesis, and the Calvin cycle, that is the synthesis part. In the light-dependent reactions, that photo part, these reactions are going to capture energy from sunlight and use that energy to make ATP and to reduce the electron acceptor NADP to NADPH. These reactions will also split water, release oxygen, and generate ATP from ADP by photophosphorylation. The light-independent reactions that are part of the Calvin cycle use both ATP and NADPH to power the synthesis of organic molecules from carbon dioxide in the air. This process is known as carbon fixation. The Calvin cycle occurs in the stroma, and this formation specifically of sugar from carbon dioxide that does require energy from ATP and NADPH can occur in the light or the dark. These reactions are light independent, so they do not require the light in order for these reactions to occur. Let's talk a little bit about light, because in order for photosynthesis to happen, we are going to need energy in the form of light from the sun. So what exactly does that mean? Now, light is a form of electromagnetic energy. Electromagnetic energy travels as waves, and we can measure these waves or the distance between the crests of waves, and we call that distance wavelength. Now, wavelength is going to determine the type of electromagnetic energy. Visible light consists of wavelengths, including those that drive photosynthesis, that produce colors that we can see. A small part of the electromagnetic spectrum consists of this visible light, and it is between 380 to 750 nanometers. Light also behaves as though it consists of discrete particles called photons. In this figure, you can see the entire electromagnetic spectrum. So it consists of many different wavelengths, most of which we cannot see. However, invisible light, which occurs between 380 to about 750 nanometers, we see visible light. So this is where visible light is on the electromagnetic spectrum. This is also where the light energy that is going to assist in photosynthesis would be found. So white light is a mixture of all the wavelengths of visible light. If you've ever seen a prism and put that prism in the light, you will see that it takes that white light and it forms a rainbow because that visible light consists of all these different colors. Pigments are substances that absorb visible light. Different pigments can absorb different wavelengths, and wavelengths that are not absorbed are reflected or transmitted. Leaves appear green because chlorophyll reflects and transmits green light. A spectrophotometer is a device that measures a pigment's ability to absorb various wavelengths. This machine sends light through pigments and can measure the fraction of light transmitted at each different wavelength. So this shows us why leaves are green, and it also shows us what the plant is going to be able to use in order to undergo photosynthesis. So the light hits the leaves, and then within the leaves we have the chloroplasts, specifically this area here referred to as the thylakoid membrane, where we are going to find that pigment chlorophyll. Now chlorophyll is green, and because chlorophyll is green, it is actually going to absorb all those colors of light except for green, which it is going to reflect back out. And that is why the leaves appear green, because chlorophyll is reflecting out that green light and absorbing the rest. And this is important because the thylakoid membrane and the pigments within there are going to use the light that's coming in to be able to have a source of energy in order to undergo photosynthesis. Now let's talk about the pigments that we are going to find within the thylakoid membrane that are going to be involved in photosynthesis. Now the main pigment is chlorophyll, but there are actually two types of chlorophyll. There is chlorophyll A, which is the main photosynthetic pigment in plants and cyanobacteria, and the only pigment that can directly convert light energy to chemical energy. It absorbs violet blue and red light. Chlorophyll B is an accessory pigment. It is the secondary light absorbing pigment, and it complements and adds to the light absorption of chlorophyll A. It can absorb violet blue and red light. There are other accessory pigments that include carotenes, xanthophylls, and other chlorophylls. Accessory pigments called carotenoids function in photoprotection. They absorb blue-green light, and they're taking in that excessive light that would damage chlorophyll itself. So what's going to happen when light comes into the leaf, gets into the thylakoid membrane, and is able to hit the chlorophyll pigment? When a pigment absorbs light, it actually goes from a ground state to an excited state, which is unstable. And so the light is going to change the state of that pigment. And when the excited electrons fall back to ground state, photons are given off, an afterglow called fluorescence. If illuminated, an isolated solution of chlorophyll will actually fluoresce, giving off light and heat. In the light-dependent reactions of photosynthesis, we are going to have what is called a photosystem. A photosystem consists of a reaction center complex, which is a type of protein complex, that is surrounded by light harvesting complexes. The light harvesting complexes are pigment molecules that are bound to proteins. They transfer the energy of photons to the reaction center. A primary electron acceptor in the reaction center accepts the excited electrons and is reduced as a result. Solar power transfer of an electron from a chlorophyll A molecule to the primary electron acceptor is the first step of the light reactions. There are two types of photosystems in the thylakoid membrane that function in the light-dependent reactions. They are photosystem 2 and photosystem 1. Now, they are named in the order of which they were discovered. So photosystem 2, abbreviated PS2, actually functions first. And this photosystem is best at absorbing a wavelength of 680 nanometers. The reaction center chlorophyll A of PS2 is called P680. Photosystem 1, or PS1, is best at absorbing a wavelength of 700 nanometers. The reaction center chlorophyll of a PS1 is called P700. During the light reactions, there are two possible routes for electron flow, cyclic and linear. Linear electron flow is the primary pathway. It involves both photosystems and produces ATP and NADPH using light energy. There are 8 steps in linear electron flow. The first step is a photon hits a pigment and its energy is passed among the pigment molecules back and forth until it finally excites P680, that chlorophyll in photosystem 2. An excited electron from P680 is then transferred to the primary electron acceptor. We now call it P680+. Water is split by enzymes, and the electrons are transferred from the hydrogen atoms to the P680+, thus reducing it to P680. P680 is the strongest known biological oxidizing agent. Oxygen is released as a byproduct of this reaction. Each electron falls down an electron transport chain from the primary electron acceptor of photosystem 2 to photosystem 1. Energy released by the fall drives the creation of a proton gradient across the thylakoid membrane. Diffusion of hydrogen ions, or protons, across the membrane drives ATP synthesis. In photosystem 1, like in photosystem 2, transferred light energy excites P700, which loses an electron to an electron acceptor. P700+, which is P700 that is missing an electron, accepts an electron passed down from the photosystem 2 via the electron transport chain. Each electron falls down an electron transport chain from the primary electron acceptor of photosystem 1 to the protein ferredoxin. The electrons are then transferred to NADP+, which reduces it to NADPH. The electrons of NADPH are then available for the reactions of the Calvin cycle, those light-independent reactions. This process also removes an hydrogen ion from the stroma. Now that we've gone through the process of linear electron flow, let's take a look at it in this figure and go through it again using this visual. So light is going to enter the leaf of the plant, and then eventually it's going to get into the thylakoid membrane and then be able to interact with these photosystems. So the first thing that's going to happen once we're inside that thylakoid membrane and out the photosystem, a photon of light is going to hit a pigment, and its energy is going to be passed from pigment to pigment molecule until it excites P680. The excited electron from P680 is going to be transferred to the primary electron acceptor. Now this P680 has lost its electrons, and what is also happening at that same time is that water is going to be split by enzymes, and the electrons that are going to be released are going to be transferred back to the P680 plus, thus reducing it to P680. And as a result of this water splitting reaction here and the electrons being transferred, oxygen is also going to be released as a byproduct. What's then going to happen here after these electrons get passed to that primary electron acceptor is that they are going to fall down this electron transport chain that is going to move them from photosystem 2 to photosystem 1, and the energy that is released by the fall is going to drive the creation of a proton gradient across the thylakoid membrane. The diffusion of those protons across the membrane are going to drive ATP synthesis. In photosystem 1, just like we saw in photosystem 2, we're going to also have a photon of light that is going to pass from pigment to pigment until that energy excites P700. That's going to cause P700 to lose an electron to the primary electron acceptor. But from photosystem 2, the P700 that's missing an electron is going to get an electron passed down from that electron transport chain to itself. Now, the electrons that are passed to the primary electron acceptor in photosystem 1 are going to fall down another electron transport chain until it gets to the protein ferredoxin. The electrons are then going to be transferred to NADP+, reducing it to NADPH. And these are going to be the electrons that are going to move into the Calvin cycle and be available for those processes. In some cases, photoexcited electrons can take an alternative path, which is called cyclic electron flow. In cyclic electron flow, electrons cycle back from ferredoxin to the photosystem 1 reaction center. Electrons are passed to a P700 chlorophyll in the photosystem 1 reaction center via the plastocyanin molecule, abbreviated PC. Cyclic electron flow uses only photosystem 1 and not photosystem 2. It is going to produce ATP, but not NADPH or oxygen from this process. So in cyclic electron flow, photoexcited electrons from photosystem 1 are occasionally shunted back from ferredoxin to the cytochrome complex and plastocyanin PC, where it is then going to go back to the P700 chlorophyll. Now, this process is going to produce ATP, but it's not going to produce NADPH or any oxygen. Let's take a look at both chloroplasts and mitochondria and their energy-producing abilities. Now, chloroplasts and mitochondria both generate ATP by chemiosmosis, but they use different sources of energy. Mitochondria transfer chemical energy from food to ATP. Chloroplasts transform light energy into the chemical energy of ATP. In mitochondria, protons are pumped to the intermembrane space and drive ATP synthesis as they diffuse back into the mitochondrial matrix. In chloroplasts, protons are pumped into the thylakoid space and drive ATP synthesis as they diffuse back into the stroma. This figure gives a very broad overall of the processes that are happening in the light reactions. So in the thylakoid membrane, light is going to cause those light reactions to occur in both photosystem 2 and photosystem 1, Water is also going to be required to be split for its electrons to be used, and oxygen is going to be released as a byproduct. We also have NADP plus gathering electrons to be brought to the Calvin cycle, as well as ATP being generated to also be brought into the Calvin cycle. So now we've talked about the photo or light dependent reactions. Now we're going to move into the light independent reactions, which are going to happen within the Calvin cycle. The Calvin cycle can be done regardless of whether or not light is present, because these are the light independent reactions. The Calvin cycle, like the citric acid cycle, regenerates its starting material after molecules enter and leave the cycle. The cycle builds sugar from smaller molecules by using ATP and then the reducing power of electrons that are carried by NADPH. Carbon enters the cycle as carbon dioxide and leaves as a sugar named glyceraldehyde 3-phosphate, or G3P. For net synthesis of one G3P, the cycle must take place 3 times, fixing 3 molecules of carbon dioxide. The Calvin cycle has 3 phases: carbon fixation, that is catalyzed by Rubisco, reduction, and regeneration of the carbon dioxide acceptor, RuBP. The Calvin cycle takes place in the stroma. It is going to use both ATP and NADPH that was generated from the light reactions in order to drive its own reactions. It's also going to require carbon dioxide, and the product that the Calvin cycle is going to make is going to be sugar, or that food source for the plant. So here is the Calvin cycle, and the Calvin cycle is going to be completed by several different enzymes throughout the Calvin cycle, and it's going to regenerate its product right here, which is RuBP, which is ribulose bisphosphate. And as these different molecules are going through the cycle, it is going to make sugar for the plant to use as its food source to generate energy from. So what's going to happen first is carbon fixation in that phase 1. So in carbon fixation, the RuBP, that ribulose bisphosphate, plus the carbon dioxide that's coming in, catalyzed by this enzyme called Rubisco, is going to make PGA, or 3-phosphoglycerate acid. Then we are going to move into the reduction phase. And in phase 2, which is the reduction phase, that PGA, or that 3-phosphoglycerate, is going to be reduced to G3P, which is glyceraldehyde 3-phosphate. This is the reverse of glycolysis. Remember, glycolysis is what we see in cellular respiration. So instead of breaking down glucose, we are actually putting together glucose. 6 carbon dioxides and 2 G3Ps are used to produce 1 molecule of glucose. And so that's what the plant is needing to make, is that glucose to be used as energy. The Calvin cycle is still going to continue, though, into phase 3 to regenerate RuBP, or that ribulose bisphosphate, and it's going to take 3 turns around the cycle to incorporate enough carbon to produce a molecule of G3P, and 6 turns to incorporate enough carbon to synthesize 1 glucose molecule. So the Calvin cycle includes these processes that are basically going to be putting these things together in order to create glucose to make that food for the plant. And because of this, it is going to require energy. The energy is going to be supplied from 18 ATP molecules and 12 NADPH molecules. What happens when the plant doesn't have enough resources to be able to do these different light dependent and independent reactions that we've been talking about? Dehydration is a problem for plants, sometimes requiring trade-offs with other metabolic processes, especially photosynthesis. On hot, dry days, plants are going to close their stomata. This is going to help to conserve water, but it also limits photosynthesis. The closing of the stomata reduces access to carbon dioxide and causes oxygen to build up. These conditions favor an apparently wasteful process called photorespiration. Photorespiration is costly because it consumes oxygen and organic fuel without producing any ATP or sugar. Thank you for watching my video on photosynthesis. I hope that it helped you to better understand the different processes that are happening within the chloroplast of the leaf of the plant. Stay tuned because I'll be making a video comparing cellular respiration and photosynthesis. And if you enjoyed this video, please make sure to like it, subscribe to my channel, and share it with your friends. If you have any questions, please make sure to put them in the comment box below. Thank you.